Hello, my name is Anna Pumo. This is Lab 2 experiment video for aseptic technique, culturing microbes for Biology 225, microbiology. I started out doing a 10% bleach solution on my counter, and then I covered it to protect it as well. I have my gloves, I have my glasses, I have my mask, my apron. I have everything set up. We have 70 alcohol, which the pipette is sitting in right now. So, and 10% bleach just available, 100% bleach for afterwards. My E. coli culture, I'm getting ready to take care of the test tube, make sure there's that there's nothing on the edge. We'll do that in a minute. And then I have my staph culture ready to go, yeast culture ready to go. And the yeast started, so 10 minutes, just started that. So in 10 minutes, that'll be ready. I'm going to go ahead and start now. So everything ready to go for this first port, the exercise 1 of Lab 2. So my pipette, so it's sitting in alcohol so that it's aseptic. Well, yeah, ready to go when I need it. So I'm going to go ahead and sterilize the culture vial tip. I also have gloves on, I think I said that. Just slowly heating the glass rim to sterilize the edges. I have a flat bottom tube, so luckily it will stay there. And then that is my E. coli tab. So here's my pipette, which I've sterilized with the alcohol. Get all the alcohol out, shake it out. And now I'm going to just make sure that all dries. .25 of the broth. In my pipette, which is hard to gauge that actually. That I could have practiced with a little bit better. Anyway, I'm going to add that to this E. coli tab, the broth. Careful not to touch the edges. That's actually going to go back into the alcohol for the next one. I would shake it vigorously. I'm going to have to be careful. My top did fall to the floor, so I won't be able to put that back on. So I will do my best to get that all dissolved well. So I'm careful not to touch the top because I don't want to add any microorganisms that are not E. coli. Now I'm going to carefully add that back in. You can see me. Well, actually, I think I need to pipette that in. I do believe that is the, yeah, Yep, yeah, okay, so that would been back into the alcohol, let that dry again. Now I'm taking the dissolved tablet from where it was initially. It's all dissolved now. I'm gonna suck it up. Careful not to touch the edges of this. Well, I really needed my top on that. Oh. Okay, I'm not sure that's gonna, hopefully I get enough to grow something. There was a lot in there. No, I'm just using this. Now this is going to be interesting to see. Feels very cross-contaminated to me. Well, potentially. So we'll use that alcohol to really clean this pipette out. Make sure there's no E. coli left in it. That will be interesting to see if that's possible. Did just lose some of my broth. That happens. Okay, that is my E. coli. That's going to be my, I'm going to carefully put this back. Maybe I'll just burn these edges a little bit more. Careful there. Okay, let that cool off. Well, hopefully that works. Shoot, good thing we use this 10% alcohol to clean up afterwards. I'm just gonna put the lid on. This is my broth culture for E. coli. It's ready to go. I'm gonna place it in my box. This is the box that I'm gonna be keeping all the cultures in. I've labeled this one E. coli culture. That's gonna go into my incubating. This is my, it'll be safe in this box, incubating for the next 48 hours. And then I'll add the staph culture and the yeast culture to this box as well. And that's going to go into a closet and sit for 48 hours. I'm actually going to I'm going to get rid of, this is going to go in my 100% bleach. This is the container for the E. coli tablet. I'm also going to move these items out. I'm going to change my gloves to use this glove. Just going to move this over. And I am going to throw this away. Well, actually, I think I'll just put it in the bleach. So all that's going to go in the bleach. New gloves. I'm going to spray this down with 10% bleach again. I did have a spill of E. coli, so I'm going to use my 10% bleach solution, clean this area up, my paper towel, and start the next part. Move on to the staph. That yeast is ready. That's my timer. So that's been sitting for 10 minutes already. So I'm cleaning up this work area, and we're going to start the staph culture. I will change my gloves again. I'm telling the surgical nurse, change our gloves a lot. Throw that away. Okay, I'm going to get another towel just to protect this area. Bring my flame back over. I'm now moving on to the staph culture. New gloves. I don't think the kit contains enough gloves for me, so I have a box, my own. hopefully this pipette with the alcohol, it sounded like we were to reuse it, just continue to sterilize it with the alcohol. So there's no E. coli here. This was just a kind of to keep me organized. So this cup is good. Just had the tablet in the container and the tablet, and that was all that was in there. That cup can be reused. So this is also my staff culture container, just to keep me organized. This is my broth with the staff labeled there. This will be my staff culture. Here's my staff tablet. And I'm going to take a look here what's happening with this pipette. Hopefully the alcohol took care of the E. coli. And I got everywhere. Okay, let's see how this one goes. I'll leave that sit for a minute. I'm going to try to keep everything up from falling. And I'm going to go ahead and sterilize the edges. Keeping this away from my face, more safety. Sterilize those edges of the glass test tube. I might actually just put this in here. Maybe that will be better. I did have a fall on the E. coli one, so I'm going to open up my staff culture tab is open now. I'm going to go ahead and take my pipette out of the alcohol. It should be sterilized at this point. I'm going to use this cup just to help me. I'm tipping over here. Have some broth in the pipette. I'm going to add it to there's some broth in the, half an ml of broth added to the pipette. I'm going to go ahead and add that to my staff culture tablet. I'm going to put my pipette back in the alcohol for sterilization, adding the top back to the tab vial. I'm going to go ahead and shake that vigorously. It now does have some broth in it. See if that'll dissolve better than the E. coli. See how that is. Hopefully that is good. I'm gonna get alcohol out of the pipette again. It should be sterilized. Now I'm going to suck up the staff culture medium or staff culture. And do my best to suck that up into this pipette. It's a lot of juice. Well, it seems like there's plenty to work with here. There's a lot coming up into this pipette. There we go. Now I'm getting it all up in there. Oh, it's splashing just like it had with the E. coli. Okay, I'm gonna go ahead and carefully add that to the broth test tube labeled staff. Not touching the edges. Doing my best not to cross-contaminate. My cat's in the laundry room. He's very upset. So that tea light's pretty hot. I think I'm gonna move my cup over here. Okay. Very careful again not to touch the edges of that test tube, of the broth tube that has the staff. Okay. Okay, that's about it for that. This is now, I'm gonna get a little bit more. Okay, I think that now, because I use my organizing container to hold that, I think there probably is now some staph in that cup, so that is going to go in the bleach. This is my staph tablet vial, that's going in the bleach. That's going in the bleach. This, my pipette's going back into the alcohol for sterilization. I'm going to just put the top back on my staph test tube. The broth, hopefully, pretty chunky. I'm not sure if that got dissolved enough. I hope so. Not sure how that goes. It'd be nice to be able to ask you some questions, but here we go. I'm going to go ahead and put this in my box. I'm going over to my incubator box holder, incubating box. So my E. coli and staph cultures are there, ready to go. I'm now moving on to the yeast culture. I'm going to throw this cup away because I believe it has that's in the bleach. I'm going to take my gloves off, switch my gloves out again. Washing my hands. Okay, one left. We're going to do the yeast now. I have new gloves. I have to get a new box of gloves. That's okay. So my yeast culture, again, this is just holding this test tube so it wasn't rolling all over. So that cup is going to be fine. Here is my yeast. It's been sitting for 10 minutes. I had mixed a half a teaspoon of the yeast provided in the kit with a 1/4 cup of warm water. It's been sitting for over 10 minutes now, probably almost 20. So I'm going to go ahead and just do the same thing as I had done with the other 2 cultures. So my pipette should be nice and clean again. Just kind of rinsing it out, sucking this up. Let's see, all the way up. Get all the top out, get all the, any of the microbes that might be sitting in the top of this pipette full of alcohol. Hopefully killing all of that or getting rid of all of that. Pipette hopefully is is clean. So I'm going to go ahead and open the yeast broth test tube that I labeled S. cerevisiae. I'm not sure if that's how you say it. I'm going to go ahead and just take the tea light and hopefully take care of any microorganisms that might be on the edge there. Now here, I forgot how much of the yeast to do, so I'm just going to look that up here. Okay, so just 0.25 ml of the yeast mixture from the cup into the broth tube. So I have a clean pipette used. Alcohol used to clean that, and I'll do my best to get a 0.25 of this yeast. Shake all that out. Let's see how much that's a lot. That's actually a lot. So this holds 5 mLs. So that's probably at least, that's at least 2 mLs. I'm just going to get rid of some of that. We're only to do 0.25. Boy, I think I almost could do like a couple drops. So I'm going to do my best not to touch the edges and just do a few drops of that yeast into this. I'm going to go back into the alcohol now. That pipette. I'm going to close that up. And there's my yeast. My yeast test tube, broth test tube. Oh, I didn't flame the lip of the broth tube before screwing on the cap. I'm just going to do that now as practice. I'm tilting that tube away from my face. Sorry, just missed that part. I think I missed it on the other one as well. So that is now sterile, and the top is going on. I'm going to extinguish the candle. The pipette is sterilizing in the alcohol right now. That is going to go into the sink. The yeast is going to go down the sink. And the alcohol. I'll be wiping down the work area with my 10% bleach solution. And I'm going to go put my 3 broth test tubes with the E. coli staph and yeast cultures and check on those in 48 hours. So here we are. Here's my incubation box. I went ahead and reused the lab pack boxes recommended or suggested. And that's it for my lab 2 experiment video of exercise 1. That's that. I'm going to go ahead and go make some agar agar plates now. That's what I'm going to do, and I don't think we're videoing that. If I'll read again. If we're not, you're not going to see me again doing that. But if I am, you'll see me doing it. Thanks. Bye bye.